All right. Hi, everybody. My name is Tyler Starr, and I am a postdoctoral fellow in the lab of Jesse Bloom at the Fred Hutchinson Cancer Research Center in Seattle, Washington. And today I'm going to tell you about a project that we recently completed on the deep mutational scanning of the SARS-CoV-2 receptor binding domain. And so before I get into that, I wanted to start with thanking a lot of the key people who also contributed to this project. As I mentioned, Jesse Bloom is my advisor. And then this work was done in close collaboration with Ali Greeny, a graduate student in the lab. And then beyond that, we had help from a couple professors of biochemistry at University of Washington, Neil King, who's a structural biologist and protein designer, as well as David Wiesler, who is a structural biologist who has actually studied coronaviruses for more than the last 6 months or so. And I also wanted to point out that everything I'm going to talk about today was recently published in this publication here. And so I'm not going to be able to go into all the details, but I invite anybody to check out this publication if you want to hear more. All right, so as you can tell from the title, the work I'm going to tell you about today is focused on SARS-CoV-2, which is this virus rendered here on the left side of the screen. And a particularly striking or notable feature of this virus is its surface glycoprotein, which is called spike. And so this is the protein that decorates the outer surface of this virus and enables it with key functional properties. The main focus of the talk today is on a particular domain of this spike protein, which is called the receptor binding domain, or the RBD. And so the receptor binding domain is this small portion of the spike protein that sits sort of buried in this kind of occluded conformation while the virus is normally circulating, but it stochastically samples an open conformation through just normal protein dynamics. And when it's in this open conformation, it exposes a surface, a contact surface, which confers specificity to binding for this ACE2 receptor. And so the ACE2 receptor is this protein on host target cells that the virus binds to, and that sets off, a chain reaction of conformational changes, which eventually allow the virus to enter the cell and initiate cellular infection. And so because of sort of the key importance of this receptor binding domain interaction with ACE2, we really wanted to get more insight into how the RBD, or this receptor binding domain, functions and confers specificity for ACE2. This RBD-ACE2 interaction has a lot of interesting evolutionary and immunological consequences. So a lot of prior work from the original SARS coronavirus epidemic, as well as ongoing work currently, shows that RBD specificity for ACE2 has a crucial role in cross-species transmission. So, for example, you can take bat viruses, which are unable to infect human cells, and if you, transplant just this receptor binding domain from a human virus onto these bat viruses, it allows them to then infect human cells. Or similarly, adapting SARS-CoV-2, for example, to replicate in mice, selects specific amino-acid changes only within the receptor binding domain. Also, because of the key interaction of this RBD with ACE2, many potently neutralizing antibodies that are known to inhibit SARS coronavirus 2 replication specifically target the RBD and prevent its ability to bind to ACE2. And so because of these key sort of evolutionary and immunological properties, we wanted to use a standard technique in the Bloom lab to study protein function, specifically within this RBD. And that technique is called demutational scanning. So demutational scanning is a experimental technique to characterize large numbers of mutations to a protein of interest, and thereby to learn more about sort of its biochemistry and evolutionary properties. The way this approach generally works is that you create a library of mutant variants of some protein of interest. And so in this case, the RBD, we're going to be creating libraries in which we introduce all possible amino acid changes across this 200 amino acid protein. What makes this demutational scanning different than just classic biochemistry is that instead of then, characterizing each of these proteins one by one, which obviously does not scale to these large numbers, you actually pool all of these mutants together and put them through some sort of experimental assay in which you can characterize each of these mutations kind of individually, but in parallel. And so in this case, we're going to be characterizing the effects of all these mutations on two properties. First of all, sort of the intrinsic folding stability of the RBD itself, as well as its affinity for human ACE2 receptor. And so at the end of this experiment, what you end up with is sort of a map of all possible amino acid changes across this protein with regards to their functional effects. And so this is just a snippet of data I'll be showing you later, just to kind of preview the way this data works. Is each row is a position in the RBD, so site 1, 2, 3. And then each columns are sites. Each row is an amino acid change. And so, for example, this blue square for introducing arginine at the second position indicates that this arginine mutation endows, for example, higher affinity for ACE2, whereas a position like this cysteine here in the fifth position with all these red squares indicates that all these mutations away from cysteine are decreasing whatever functional property is shown here in this heat map. So that brings me to kind of the specifics of what we did here in this current study in which we undertook one of these demutational scans for the SARS-CoV-2 receptor binding domain. So I'll start by explaining in a little bit more detail the specifics of our experimental assay, and then we'll go through some of the observations we made from this data, including what it teaches us, a little bit about the structure-function relationship of how this receptor binding domain works. We'll look a little bit at how these maps of mutational effects can help us to understand currently circulating genetic variation within the SARS-CoV-2 pandemic. And then finally, I'll touch on what our maps can teach us about the epitopes on the RBD that are targeted by neutralizing antibodies. All right, so to kind of do this deep mutational scan in a high throughput setup, we turned to a classic system from protein engineering and biochemistry, which is yeast surface display. So yeast surface display is a technique that allows you to express some protein of interest on the cell surface tethered to individual yeast cells. And so in this case, we have yeast cells which have receptor binding domain multivalently expressed on their cell surface. And when it's there, we can do two key things. We can use fluorescent detection of a tag on the RBD itself to probe how well expressed the RBD is on an individual yeast cell surface. And then beyond that, we can incubate with soluble ACE2, which is also fluorescently tagged, so we can monitor how well any given RBD variant binds to ACE2. So to kind of show what this looks like, for example, if we just take the SARS-CoV-2 RBD and express it in its wild type form in yeast, we can then do a titration series across an increasing concentration range of ACE2, and we can monitor the amount of ACE2 binding via flow cytometry. And so what you can see here is that as we increase the concentration of ACE2, we get sort of a dose response curve of ACE2 labeling of our yeast cells. And so from this relationship between binding and concentration, we can fit these titration curves, which are sort of these classic biochemistry curves from which we can compute the dissociation constant, or the KD, which is a measurement of the strength of biochemical interaction between, in this case, the wild type SARS-CoV-2 RBD and human ACE2 ligand. So to transition to doing this in more of a high throughput format, we first needed to create a mutant library of SARS-CoV-2 so we can characterize the effects of all these mutations on this ACE2 ligand affinity, as well as the kind of expression level of the RBD on the cell surface. And so to do that, we constructed duplicate mutant libraries using a PCR-based codon mutagenesis technique, which yielded us libraries of mutant variants. We also appended these barcode sequences downstream from the individual variant RBD genes. And then we can put this system of this library of mutant variants into our yeast display system. But in contrast to what I showed you before, instead of it just being a single genotype, each individual yeast cell in the population will now have a different RBD variant on its cell surface. To kind of allow us to see what's happening in an individual cell, we use these barcodes then. What we do is we use long-read PacBio sequencing to link individual barcodes to the actual mutant variants that they're associated with. And what that means is for any downstream experiment, we can just do short-read Illumina sequencing of the barcodes. And so, for example, if we read this yellow barcode, we know it represents this individual single mutant genotype. And then I'm showing you here just one table illustrating the kind of coverage that we get in these libraries. And if you look at the second column, you can see that in each of our duplicate libraries, we have very high coverage. We get virtually every single possible amino acid change to the RBD present in our libraries. I think we're missing only 15 of the almost 4,000 different amino acid changes that are possible to make within this domain. So next, we wanted to kind of do these titrations, but in high throughput. And so what I showed you before, once again, was just doing a titration for sort of an isogenic yeast population. But as I was alluding to in the last slide, now we have a mixed library population in which each yeast cell has a different genotype on its cell surface. And so if we look at this gray dose response curve here, we no longer have just a single response curve because each genotype in the library is sort of responding to the ACE2 labeling concentration on its own sort of dissociation constant driven titration curve. And so rather than just monitoring fluorescence via flow cytometry, what we do for the library experiment now is we use fluorescence activated cell sorting, or FACS, which is a flow cytometry based method that allows you not only read out fluorescence, but also separate cells on the basis of the fluorescence signal. And so what we can do is at each of these ACE2 concentrations, actually sort the cells into these 4 bins, illustrated by the dotted lines. And then we use Illumina sequencing to read out which yeast genotype is found in each of these bins across each of these ACE2 concentrations. And so, for example, a genotype like perhaps the yellow, which has high affinity for ACE2, will begin to kind of fall into the bin 2, 3, and 4 partitions at relatively moderate ACE2 concentrations. That might be different from a genotype like the blue genotype, which perhaps does not bind ACE2, which might be found in bin 1 across this entire concentration range. And so when we do this sort of FACS coupled with deep sequencing, what we end up getting are these titration curves, but for each individual variant in the library in parallel or simultaneously. So what I'm showing here on the right are just a random selection of genotypes in our library, which are labeled on the top. And then from this kind of massively parallel experiment, we have the titration curve for each of these variants sorted independently. As you can see how each genotype in the library is able to have its own kind of variant-specific titration curve from which we can compute this dissociation constant, and therefore end up with a measurement of the effect of each amino acid on this ACE2 binding affinity. And then as our last sort of experimental measurement, beyond simply determining mutation effects on ACE2 binding, as I alluded to before, we're also measuring the effects of these mutations on the RBD expression level, which is a correlate of sort of the folding stability of the RBD. And so this is a similar technique. It's FACS coupled with sequencing, but instead of doing it across an entire dose response range, we're just doing it, one time at one labeling of RBD expression levels. So we sort cells into bins based off of their level of labeling for RBD expression level, and then we use sequencing to figure out, which genotypes are, higher or more highly or more lowly expressing RBD on the cell surface. All right, so that's the technique. There's a whole lot of QC that goes into this, and we also did a lot of validation, which I'm not going to get into today, but I do encourage you to check out the paper to see kind of the reliability of these measurements. But what we end up with are these heat maps, as I showed you very small snippets of earlier, illustrating the effects of all amino acid changes at each position in the RBD on these 2 biochemical phenotypes. So on the top 2 panels are all of the RBD sites sort of annotated by the mutation effects on expression, that sort of stability metric. And then on the bottom are all of these mutation effects on the ACE2 binding affinities. And so obviously these are very detailed maps, and I don't expect you to necessarily take away too much from looking at them quickly from a bird's eye view, but they are a super rich kind of display of a lot of important information. And so one key thing we wanted to achieve with this project was to make sure that any researcher doing who knows what about SARS-CoV-2 that might be able to leverage this information also has the ability to kind of engage with these heat maps. And so I'll flash up the link here, but it's also present in the paper. We made interactive versions of these heat maps, so anybody can kind of open them up on their own and gaze at them for hours and hours, just like I did, to kind of emerge with some key insights. But just to kind of give some of this bird's eye view, I'll focus on a few patterns that we can see in these heat maps. So in these heat maps, as I said before, red indicates mutations which are deleterious for a phenotype, such as stability or binding. And then white indicates mutations that are neutral, and blue are mutations that enhance that biochemical property. And so kind of as you expect from just classic protein biochemistry, most mutations to this RBD do indeed destabilize the protein or inhibit its ability to bind ACE2. But at the same time, we do see many positions where lots of mutations are neutral. And perhaps even more surprisingly, there are quite a few positions where a lot of blue mutations are cropping up, which is to say mutations that would enhance either the stability of the RBD or even its affinity for human ACE2. And so we'll revisit some of those specific mutations a little bit later on. Beyond that, we can also see heterogeneity in the level of constraint on different positions across the RBD. So there's some positions where, any mutation is kind of more or less tolerated the same, whereas some positions are highly constrained where any mutation away from the wild type amino acid is deleterious for function. We can also see that, if you kind of squint your eyes, the positions that are kind of constrained for expression and binding tend to overlap, although there are some regions where there's kind of increasing constraint for expression or binding individually. So with that sort of bird's eye view, we'll move on to talk a little bit about some more specific sort of findings or observations we made about the relationship between sequence structure and function within the SARS-CoV-2 RBD. So here what I'm doing is taking this heat map, sort of the average mutational tolerance of each position, and mapping it to the structure of the RBD bound to ACE2. And so here we've annotated a few different features of the RBD. We've annotated what we're calling a core RBD, which is the central beta sheet folded core of the protein, which is sort of the scaffold on which this RBM, or the receptor binding motif, sort of loopy region sits. Beyond that, I'm also showing some disulfides in yellow. And then we're showing the bound ACE2 ligand in dark gray at the top. And so each sphere on the RBD here is colored by the average mutational effect on the stability of the RBD. And so sort of as you'd expect, a lot of the positions that are most constrained with respect to mutational effects on stability are sort of buried in the core of the protein. They're these sort of internal positions where if you muck them up with mutation, you end up destabilizing the protein. We can do the same thing, coloring these spheres now by their mutational effects on binding. And as I pointed out before, we see some overlap. Many of these kind of buried core positions are also constrained with regard to their effects on binding. But beyond that, we can see that many more of these positions that cluster up at the interface with ACE2 are constrained with respect to binding, as you'd expect. To look at this in a little bit more detail, I'm showing here now for each site the correlation between its sensitivity to mutation with respect to binding and expression. And so once again, you can see that most positions sort of follow a general trend where if they're sensitive for stability, they also lose binding. And then of course, you can see there are some positions where they are not particularly sensitive with respect to mutation effects on expression, but they are for binding. And when we look at these on the structure, these are indeed these positions that cluster to the ACE2 interface and sort of interact with these two hotspot residues on ACE2, these two lysins. We actually saw a little bit kind of more interesting pattern in this regard, taken even to the more extreme, where we see that there's actually, at some positions, sort of trade-offs between mutational effects on stability and binding. So here I'm showing heat maps focused just on 5 positions that illustrate these trade-offs. And what you can see at each of these 5 positions is that many of the mutations on the left-hand heat map have deleterious effects on binding, even though they would have improving effects on stability or the expression of the RBD. So one example of this is glycine 502, where any mutation severely hinders binding, even though many of these polar amino acids would enhance expression. And when you look at the structure, this makes sense. This glycine 502 on the RBD is in very, very close structural proximity to this important beta hairpin on ACE2. And there's simply just not room for a side chain to accommodate ACE2 binding. So even though putting a side chain here might enhance stability, it just wouldn't work for ACE2 binding. For these other 4 sites, we saw that these are hydrophobic residues that are exposed on the RBD surface. And so exposed hydrophobics are known to destabilize proteins and contribute to things like aggregation. So mutating these hydrophobic residues would enhance expression, but these hydrophobic residues are important for packing at the ACE2 surface, which is why they have this binding specific constraint. All right, so this has been focusing mostly on constraint, but I also did show you that there are many kind of other interesting enhancing mutations with respect to stability and binding. And so I'm going to focus first on stabilizing mutations. And in particular, we were motivated by this idea that there's a lot of people who want to engineer RBD protein to have certain properties such as immunogenicity or stability. And these maps might be useful for that. And so here's one example of a recent preprint, which is using an RBD-based vaccine, where it displays these RBD proteins on the surface of nanoparticles. Another kind of RBD-based tool are these molecular probes that people can use to isolate B cells or antibodies that target the RBD of SARS-CoV-2. And we figured our maps could be useful in two regards here. We could be making RBD proteins that are just better behaved biochemically, for example, more stable or more highly expressing. But beyond that, as we learn more about, for example, antibody epitopes on the RBD, our maps could be useful for resurfacing these RBD proteins to, for example, hide epitopes where perhaps we don't want antibody responses, or to make better molecular probes to isolate specific antibodies. So to look at this a little bit more, I'm showing once again the map just for mutational effects on expression. And in particular, we were compelled by this one region here, kind of down by amino acid 355 to 385, where we see many blue mutations, so mutations that enhance stability. And so once again, this is, of course, our yeast display assay. So we did want to validate whether these really have a stabilizing effect. And so in collaboration with Dan Ellis and Neil King, we cloned individual point mutants of the SARS-CoV-2 RBD and expressed them in mammalian cells. And they saw that these individual mutations, so individual single mutations, can enhance the soluble expression of RBD up to 5-fold. And then on the bottom plot, we see that this is also accompanied by increases in the thermal stability of the RBD. And so we found it super interesting that these, mutations that we found really have big improving properties on the general expression and stability of isolated RBD protein. And we have some interesting ideas about why this is the case, which if anyone's interested in, I'd love to talk about further. Beyond that, we also noticed in our data that we have many positions where we knocked in N-linked glycan motifs. So those are NXST mutations, which are shown in boxes on this heat map. And at many of these knock-ins where we add S's or T's to positions after an asparagine, we saw that that would actually enhance expression. And so N-linked glycan knock-in is a common way to resurface, probes or vaccine immunogens. And then we see a bunch of positions across the RBD where these glycans can be tolerated. Coming back to now the binding effects of mutations, we're zooming in now on the heat map just of positions that are actually at the ACE2 interface. And sort of in contrast to many proteins where the key functional sites are especially constrained, we were actually surprised to see that there is a lot of mutational flexibility at these positions at the ACE2 interface, where many mutations are tolerated. And as I said before, there's actually many mutations that actually enhance the affinity for ACE2. We were particularly interested in 3 positions that cropped up here, 493, 498, and 501, where there are many of these strong affinity enhancing mutations. And when you look at the actual molecular or atomic interface between the RBD and ACE2, these residues are kind of involved in these networks of polar contacts with key ACE2 residues. And so just looking at the structure alone, you might not have been able to predict that many of these mutations would be tolerated or enhance affinity, particularly because many of these kind of blue mutations are actually hydrophobic mutations, which would be removing these polar contacts entirely. And so I think that shows there's a lot of both mutational, but kind of correspondingly structural flexibility in how this interface can be kind of engineered or rearranged. And that's reflected in the evolutionary record in which this interface and this RBD in general is actually quite variable among kind of SARS-related coronavirus isolates. All right, so next we wanted to know whether we could use our maps to understand more about sort of the evolutionary forces that are guiding SARS-CoV-2 genetic variation at this current phase of the pandemic. And so this pandemic is interesting from one regard in that we have kind of new genomic technologies that have really come online and really changed the way that we can understand genetic variation in this emerging pandemic. And so I think currently there's something like 75,000 individual SARS-CoV-2 genomes that are available. And so back when we did this analysis in May, it was something like 30,000 mutation spike sequences available. We looked at what variation is present in these sequences within the RBD. And in general, we see that RBD mutations are still quite rare among sequenced SARS-CoV-2 isolates. But beyond that, besides just seeing their frequency, we now also have functional annotations of these mutations effects. So what I'm showing here is on the y-axis, the effects of mutations on binding, and then we binned mutations by the number of times they've been observed on this repository of sequenced SARS-CoV-2 isolates. And so what you can see is that for any mutation that has been seen, more than a handful of times, there's this increasing bias in what types of mutational effects are present among those mutation sets. And so anything, seen more than 6 times has essentially 0 effect on binding. And so we see that as evidence for purifying selection, in which mutations that would decrease binding are being selected against, and that's why they're not being seen at high frequency among these sequenced variants. Beyond that, as I pointed out to you, we have many affinity enhancing mutations as well. And so these mutations are indeed accessible to the wild type virus. They can be accessed by single nucleotide mutations, but they're not being fixed or even seen in natural SARS-CoV-2 circulating strains. And so we thought that was interesting. Given that these mutations are available but not being seen, we take that as evidence that perhaps there's not any selective pressure for higher affinity for ACE2, which is to say perhaps the virus is already sort of in that Goldilocks sweet spot for the degree of affinity that it has for human ACE2. Kind of consistent with this idea that there aren't really strong phenotypic consequences of mutations that are currently circulating in the RBD, we did look at sort of the most prevalent mutations in sequenced SARS-CoV-2 isolates. And so the top 5 prevalent mutations are shown here. And you can see once again that we're really not seeing any strong biochemical consequences for these mutations with respect to our phenotypes of ACE2 binding and expression, with just the one exception being this V367F mutation, which we do ascribe a large stability enhancing effect. And this was indeed one of the mutations that we validated in the purified RBDs. And so we don't know what the consequence of that is for the actual virus, but I do think that is quite interesting to note and perhaps is worth following up on. And then, of course, I think, it feels like it's been a really long time, but it seems like we're actually probably still in quite an early phase of this pandemic, at least from the perspective of the virus and its evolutionary dynamics. And so as this virus continues to circulate in the population and begin to encounter things like immune pressure and, a less wide availability of susceptible hosts, it'll be interesting to see or continue to surveil mutations that are arising in the context of these maps. All right. And then last, I'll talk about what our maps might be able to teach us about antibody epitopes within the RBD. So as I mentioned at the beginning, the RBD is a common target of antibodies that neutralize SARS-CoV-2. And these antibodies, I'm just showing a structural alignment of several of them, sort of decorate a bunch of different surfaces on the RBD. And related to this question about how it might evolve over time, one super important question that we don't really know much about yet is whether this type of virus, or whether this virus will be able to do this type of evolution similar to how flu evolves to sort of turn over its antigenic determinants over time, or whether it will be more sort of antigenically stable, such as a virus like measles. So to begin to get some insight into that, we were motivated by a paper that came out also during the summer, which passaged viruses in the presence of monoclonal antibodies, and then sequenced to see what types of mutations it was fixing to escape these neutralizing antibodies that they were grown with. And so even though these antibodies target the regions that surround these kind of mutationally constrained contacts for ACE2, the sites at which the antibody escape mutations were actually accruing in this study were kind of finding the mutationally tolerant residues in this otherwise constrained region. And the specific mutations that we're fixing, shown on the right, as you'd expect, have sort of biased, more neutral effects on ACE2 binding affinity, obviously because if you need a mutation to escape an antibody, but you also lose ACE2 binding, the virus wouldn't be able to tolerate that. So to look at that in a little more detail, I'm showing our kind of mutational constraint coloring on the surface representation of the RBD. And then we can outline residues that participate in epitopes or interaction interfaces. And so for reference, the black outlines in this image here illustrate which residues contact ACE2. And as I showed before, these are quite mutationally constrained. And then we can show different antibodies that target the RBD and sort of the regions that they interact with. And so on the top, you can see many antibodies sort of target one or two of the same patches that interact with ACE2, although they also sort of extend their structural interfaces to other less constrained regions. And so this suggests that there are multiple individual epitopes that can be targeted that overlap these constrained ACE2 contact positions, but at the same time, it might be beneficial to achieve better so-called epitope focusing on specifically the very constrained positions that make these exact ACE2 contacts. On the bottom, we can see a little bit more distant epitopes in this core RBD, which are constrained with respect to mutational effects on expression. And so overall, this kind of structural mapping highlights the presence of multiple kind of mutationally constrained patches on the RBD surface. And it might be important to consider the ability of, for example, vaccine-induced immunity to elicit antibodies to multiple of these patches simultaneously to perhaps prevent the ability of virus to evolve to evade antibody-based immunity. Finally, we also did note there was one surface or one patch on this surface where we don't know of any antibodies yet that have been structurally mapped, but does show high intrinsic mutational constraint. So this patch is sort of centered around this residue E465. And so we think it's interesting to kind of guess whether there will eventually be antibodies that target this region and whether this might be a sort of mutationally intolerant or constrained region that can't evade such antibodies. All right, so I think that's everything I want to talk about today. Like I said, there are a lot more details in the paper that I couldn't get into, but I wanted to show some of the kind of early observations we've made from this sort of mapping of mutational effects on the SARS-CoV-2 RBD. With that, I wanted to thank, once again, all the people who contributed to this work, as well as the funding, and once again emphasize that if anybody is doing work on the SARS-CoV-2 RBD and thinks that these maps of mutational effects could be helpful, I really encourage you to check them out, and please don't hesitate to reach out to us. We would love to help anybody doing anything to be able to take advantage of this resource that we generated. So thank you very much.